Proteins play a pivotal role in the proper functioning of any organism. The synthesis of proteins occurs in the cell in two phases, namely transcription and translation. Transcription takes place in the nucleus, where the DNA acts as a template for the synthesis of messenger RNA. It is a process by which genetic information in DNA is transferred to a molecule of messenger RNA. It is governed by the principle of complementarity, with one exception, that adenine now forms base pair with uracil instead of thymine. Messenger RNA carries the genetic information to the cytoplasm for protein synthesis, and hence it gets the name messenger. The transcription process is different from DNA replication. In replication, the total DNA of an organism gets duplicated, while in transcription, only one DNA strand gets copied into RNA. In case both DNA strands act as templates in transcription, two types of proteins will be produced. One with the correct sequence and the other with the reverse sequence of amino acids. Moreover, the simultaneously produced RNA molecules would become complementary to each other and will form double-stranded RNA. As a result, RNA will not get translated into protein and the process of transcription will get nullified. However, such an occurrence is quite rare. The segment of DNA which takes part in the transcription process is called the transcription unit, and it encodes for at least one gene. Let's understand the transcription unit in detail. It consists of three main regions, namely the promoter, the structural gene, and the terminator. The promoter is the starting point of the transcription process and is present upstream at the 5' end of the structural gene. In the promoter, there is a binding site for RNA polymerase, which moves along the DNA strand from 3' to 5' direction. With the help of RNA polymerase, the promoter can code for the two strands of the DNA helix. However, these two DNA strands will get reversed if the promoter switches its position with the terminator. The next part of the transcription unit is the structural gene, which consists of two DNA strands having opposite polarity. The strand with the 3' to 5' polarity acts as a template and is called the template strand, while the DNA strand, which has 5' to 3' polarity, is known as the coding strand and is displaced during transcription. The sequence of this strand is the same as that of RNA, except that thymine exists in place of uracil. The structural gene can be defined in terms of a cistron or segment of DNA that codes for a polypeptide. The structural gene in a transcription unit can be either monocistronic or polycistronic. A monocistronic structural gene contains the genetic information to translate only a single protein and is found in eukaryotes. On the other hand, a polycistronic structural gene carries the information of several genes which can be translated into several proteins. It is found in prokaryotes. The monocistronic structural gene has a split gene arrangement, where sequences called introns and exons are present. However, in this gene arrangement, only the exons act as coding sequences. The process of transcription helps remove introns and joins exons as functional messenger RNA. It is the exons in the structural gene, along with the promoter, which determine the inheritance of a character. The structural gene also consists of regulatory sequences, which regulate the functions of other genes but do not code for any RNA or protein. They are also known as regulatory genes. At the 3' end of the structural gene is the terminator, which is another important part of the transcription unit. The terminator defines the end of the process of transcription. Transcription also happens to be the first stage in the process of gene expression. As it helps in the transcription of a gene into messenger RNA and encodes for a protein.